Good afternoon, everyone, and welcome to this month's IDDD Roundtable Workshop. We're really delighted to have a couple of our colleagues from here in Houston who are experts nationally and internationally in this space, and just really delighted that they could join us today. I know if you received the invitation to this, you received a couple of short bios, but I'd like to just kind of touch on some of those highlights before we get started. As we welcome, first up today will be Dr. Kyoji Tsuchikama, who is an assistant professor at UTHealth since 2014. He earned his PhD in organic chemistry in Japan and did his postdoc at the Scripps Research Institute. Since coming to UTHealth in 2014, he's been leading a medicinal chemistry lab as the principal investigator. His research programs are supported by NIH, DoD, and CBRIT, and his primary interest is in developing linker technologies for generating antibody drug conjugates as cancer chemotherapeutics. And so after we hear from Dr. Tsuchikama, we're going to hear from Dr. Zhiqiang, who is a professor of molecular medicine and the Robert A. Welch Distinguished University Chair in Chemistry and Director of the Texas Therapeutics Institute at UTHealth. Where his lab focuses on antibody drug resistance mechanisms, biomarkers for therapeutic antibodies, and antibody drug discovery targeting human disease. He also directs the Therapeutic Monoclonal Antibody Lead Optimization and Development Core facility that's funded by CBRIT, and has served as a Chief Scientific Officer at Epitomics Incorporated and was Director of Biologics at Merck. And so with that, just a little bit of kind of the housekeeping for today's workshop. We'll hear first from Kyoji, and then we'll hear from Zhiqiang. You're welcome at any point during the workshop to enter your questions in the chat, but we will have a dedicated roundtable Q&A session after both of their talks where we'll just have some informal discussion where they can address your questions. But again, feel free to enter those in the chat at any time, just so you don't forget it later. And with that, I'll turn it over to Kyoji.
Thank you for the detailed introduction, and thank you for giving me this great opportunity to talk about my recent research projects and achievements. Today, I'm going to talk about our chemical linker technologies for generating truly effective ADCs for mainly cancer therapy. So let me share the slide. And now it's in the full screen mode. All right. So as most of the people here know, cancers are treated by several different clinical options. The primary option is surgical removal, but in most cases that comes with other therapeutic modalities as monotherapy or combination with surgical removal, which is radiation, molecularly targeted therapy, and more recently, immunotherapy using immune checkpoint inhibitors. And chemotherapy has been one of the major clinical options for cancer treatment. And as a new addition in this category, ADC, antibody drug conjugates, have been developed and used in blood cancers and solid tumors. And ADCs have shown remarkable, successful clinical outcomes in the recent years. Why is ADC really effective and promising in cancer therapy? As you can see in the left side, conventional chemotherapeutic agents are small molecules. And most small molecules used as chemotherapeutics is often doesn't have cancer cell specificity. That means it can kill not only cancer cells, but also other normal tissue and cells. And that leads to adverse effects. And because of its short circulation life in the body, patients need to be administered frequently, like every day or every other day. In contrast, antibody drug conjugates deliver highly potent cytotoxic molecules called payloads and conjugated to the monoclonal antibody structure. By combining the antibody molecule and small molecule chemotherapeutic agents, we can achieve target-specific site killing effect thanks to the cancer cell specificity derived from the mAbs structure. And that often results in broader therapeutic window compared to small molecule-based drugs. And most ADCs have much longer circulation life than do most small molecule-based drugs. So these benefits greatly help this drug class shows much improved clinical efficacy. And as of right now, we have 9 ADCs that have been approved by FDA for blood cancers and solid tumors. And more than 100 ADCs are currently tested in clinical trials in different phases. These numbers clearly show this category is a very exciting one in the cancer drug development. Let's take a look at the structure of the ADCs. And as you can see here, ADC molecule consists of 4 major components. First one is, of course, the most important part, monoclonal antibody against a specific cancer-specific receptor. Most ADCs are derived from humanized or fully human monoclonal antibodies to minimize the immunogenicity issue. And payload or cytotoxic drug molecules are conjugated through stable chemical linkers. And this linker may or may not have a specific cleavage mechanism to release the payload component inside the cancer cell. Depending on the cleavage mechanism, the linker stability varies. And that is a very important part, because if the linker is cleaved during circulation in a premature manner, if it undergoes premature degradation, the ADC molecule loses the payload before reaching to the tumor. So that results in undesired systemic toxicity or poor efficacy. And the linker payload module is connected to the antibody structure through various conjugation chemistry. The standard methods include lysine coupling, cysteine alkylation, and enzymatic reactions. So depending on the choice of each component, each ADC has a totally different biophysical and physical, chemical, and biophysical properties. So optimizing each component is very critical to obtain clinically promising ADC molecules. As you can see here, most ADCs go through a very simple mechanism, and eventually that releases the payload inside the cancer cells. First step is to the complexation between antibody molecule and its target receptor. That eventually promotes the internalization through anesthetic pathways or other unknown pathways that is still being studied by many labs. And that complex is eventually delivered to lysosomal component and undergo degradation. And finally, the conjugated payloads are released inside the cancer cell. So the process, this pathway is not really complicated, but it involves multiple steps, and each step significantly affects the overall efficacy of ADC molecules. That's why these 5 or maybe more factors need to be optimized at the same time to achieve truly effective ADC-based treatment. Another very important parameter is the homogeneity of ADC molecules. Most conventional methods provide heterogeneous ADCs, meaning ADC consists of various species that differ in the conjugation site and or the number of payload molecules conjugated to the antibody structure. This distribution provides complex PK/PD profiles, and producing such ADCs needs to be strictly controlled to assure the high product quality. And that is another factor that needs to be fully optimized before this type of ADC goes to enters the commercialization step. In contrast, recent researchers have recently developed different methods, conjugation methods called homogeneous conjugation. Using these type of conjugation methods, now we can produce a single ADC product consisting of a single defined drug to antibody ratios and defined conjugation sites. Such a single pure product usually gives simple PK/PD profiles. And thanks to the site-specific conjugation methods, the production is more facile and consistent. And many of recent studies have shown that homogeneous ADCs usually show more improved efficacy and safety compared to heterogeneous ADCs. So site-specific conjugation is another key factor for successful antibody-drug conjugation. Based on this background, my lab has been committed to developing novel ADC-linked technologies. Today, I'm going to talk about 2 different technologies. First one is branched linker. Most conventional ADC linker can accommodate only 1 payload per linker structure. And most conventional methods provide heterogeneous ADCs, as I explained in the previous slide. Our branched linker ADC branched linker technology enable incorporating 2 or more payload molecules at the same time. And this increased number of payloads directly affects the potency of ADC molecules. Another technology I'm going to talk about is glutamic acid, valine citrulline, or gluvalicid enzymatically cleavable linker. Most ADCs contain enzymatically or cleavable linker called valine citrulline. But valine citrulline is known to be unstable in rodent plasma. And to overcome that problem, my lab has modified the structure by adding another amino acid next to that valine citrulline sequence. And that dramatically improved the plasma stability, but at the same time, this sequence allows for quick payload release inside the cancer cell, leading to improved efficacy. The branch linker can be easily installed onto any type of antibody molecules, as shown here. First, a very small branch linker containing 2 AZ groups is installed onto the side chain of glutamine 295 using microbial transglutaminase. This reaction is site-specific, and the resulting antibody linker conjugate has the linker structure only at this glutamine 295 or Q295. After that reaction, the DBCO MMAF payload module is added to that mixture. And the reaction between DBCO on this structure and AZ is very selective, and neither of these chemical functional groups neither of these groups react with other side chains of the amino acids. So thanks to that site specific reactivity, this sequential reaction provides antibody drug conjugates with defined drug to antibody ratio. In this case, it's 4. And the ADC we generated showed improved efficacy in HER2 positive SKBR3, MDA, MB453 cells, while conventional linear linker-based ADC showed less effective cell killing potency because of the lower drug to antibody ratio. In this case, it's about 2. But the increased DAR didn't promote the non-specific cell killing, which was proved in the evaluation using HER2 negative cell line, MDA MB231. So our branch linker could simply improve target antigen specific cell killing potency. So we validated the effectiveness of the branch linker strategy, but one serious problem was not solved at that stage. That is the instability of the valine citrulline dipeptide enzymatically cleavable linker structure. So that was not a problem in vitro assays, but in mouse-based studies, valine citrulline dipeptide linker is known to quickly degrade and release the payload at a very fast rate. As a result, efficacy and tolerability data are negatively impacted. This is not a problem in human because human plasma doesn't have such an protease that can cleave this valine citrulline sequence. Only cancer cells, the lysosomal enzymes inside the cancer cell can cleave this peptide. But if we have this instability issue in mouse models, we can't smoothly bring up a promising drug to the next stage using advanced models such as primates. In other words, if we see any negative results in mouse models, we can confidently bring that such ADC candidate to the next level. To overcome that problem, we attempted to modify the structure by adding one more amino acid next to the valine citrulline diamino dipeptide sequence. We screened a variety of amino acids, and then eventually we found that glutamic acid E or aspartic acid D dramatically improved the half-life of the valine citrulline dipeptide-based sequences. As you can see here, the half-life is almost more than tenfold longer than the conventional valine citrulline linker. So we found acidic addition of an acidic amino acid to the valine citrulline linker is a simple but powerful solution to overcome the instability issue in mouse plasma. Encouraged by the result, we tested the efficacy and pharmacokinetics profiles and efficacy in mouse models. As you can see here, our EEV-CYT-based ADC didn't show quick payload. As you can see here, the EEV-CYT-based ADC didn't lose the payload and actually this curve completely matches the I didn't show that kind of curve in this figure. I'm sorry, but this curve totally matches that of unmodified mAb, meaning payload molecules were completely retained onto the linker structure. In contrast, conventional valine citrulline-based ADC quickly lost the conjugated payloads. And this makes a big difference in treatment efficacy. As you can see, these two test models, our EEV-CYT-based ADC showed significantly remarkable treatment efficacy only with a single injection at 3 mg/kg. The purple curve is a lower dose, 1 mg/kg, and it still showed some efficacy. In contrast, conventional valine citrulline linker could not treat this tumor model as well as another HER2 positive tumor model with just a single injection. So this result clearly demonstrates the EEV-CYT technology assures the plasma stability as well as maximal efficacy in mouse models. And this means we can confidently use or advance this ADC to the next level for future clinical trials. So let me explain another critical issue in the cancer therapy. That is drug resistance and heterogeneity. As you know, most tumors consist of multiple different types of cells, and some cells may express antigen or the target antigen, but some of the neighboring cells may not express such target antigens. In this situation, if you use an ADC targeting one antigen, it may be able to kill that positive cells, but the negative cells eventually grow back and becomes a dominant species, and that is a part of the reason of acquired drug resistance. To overcome this problem, there's one solution, that is the payload with bystander effect. Some payloads are very hydrophobic, and they can kill not only the initial targeted antigen positive cells, but also the neighboring antigen negative cells by diffusion from the initial cells. Hydrophobic molecules can easily penetrate the membrane, so that is how such payloads with hydrophobicity, high hydrophobicity can kill neighboring cells. That effect is called bystander effect. But one problem is such hydrophobic payloads can be easily pumped out cells that express PGP or other drug-resistant proteins at higher levels. So that's a dilemma of the use of such hydrophobic payloads with bystander effect. To overcome that problem, we evolved our branch linker technology so we can incorporate not just a single drug, but two distinct drugs at the same time. We call this dual conjugation or dual conjugate. By incorporating two different drugs, we can such a molecule can exert on dual modes of action, and eventually that strategy may overcome drug resistance and cancer heterogeneity issue. To prove if this strategy works, we decided to install two different drugs. One is payload with bystander effect, and the other drug is a more hydrophilic payload that does not show bystander effect, but it's not pumped out by PGP positive or resistant cells. The left panel is each step of linker conjugation and payload installation, characterized by mass spectrometry. As you can see here, our conjugation provides a single pure product at each step. And in the last step, two different payloads were installed in a stepwise manner. And we successfully obtained dual drug ADC with the drug to antibody ratio 4+2. In this case, 4 is mAe payload, and 2 green payloads were mMAF, another payload. And gratifyingly, this dual ADC showed cell killing efficacy in this low HER2 tumor model with MDR1 positive, MDR1 expression. This is very drug resistant to many types of ADCs. As you can see here, even in HER2, one of the FDA-approved ADCs couldn't kill this cell line. So this data demonstrates the high potency of our dual drug ADC. And we also tested the efficacy of our ADC, a dual drug ADC, in heterogeneous breast cancer model. We prepared a tumor-bearing mice consisting of HER2 positive GYM1 and HER2 negative MDA-MB231 cells as an admixed model. So this study clearly demonstrated that the blue curve, which is our dual drug 4+2 ADC, showed the best treatment efficacy compared to single drug MMA4 ADC, or even a co-injection of MMA-E ADC plus MMA-F ADC, 1-to-1 mixture of those two different ADCs. So this data clearly demonstrates that dual drug ADC strategy is a preferred way than compared to co-injection of two different ADCs. And we also have confirmed that the tolerability of this ADC, dual drug ADC, is pretty high. Even at 40 mg/kg, we didn't see any symptoms or toxicity in mouse models. That is much higher tolerability than that of TDM1. Okay, so in conclusion, our ADC linker technologies have shown the remarkable dramatic improvement of the linker stability in plasma, and it can maximize the ADC efficacy and safety profiles. We believe the use of this linker technology, as well as the branch linker dual conjugation strategies, will provide a very promising panel of promising ADCs for future clinical studies. Okay, this is I really appreciate the tremendous support from our collaborators and lab members, and in particular Dr. Anami and my wife, Dr. Tsuchikama, Chisato Tsuchikama, conducted most of the research projects I presented. And all 80 monoclonal antibodies were generated in the next speaker's, Dr. Zhiqiang's lab. And I really appreciate their support for this project. Thank you. And I'm happy to answer your questions. Not now, but in the Q&A session.
Thank you, Kyoji. That was a great talk. I'm sure we'll have a few questions. Again, feel free to be jotting those down in the chat or just making notes so that you can remember to ask them in the roundtable portion. And so with that, we'll turn it over to Dr. An. You're muted still. Dawn, can you unmute? Okay, there we go.
Okay, can you hear me now?
Yes.
Can you see my screen?
We can. It's not on the first slide and it's not in presentation mode yet.
Mm-hmm.
Perfect. You're good to go.
Okay. So thank you, Suzanne, again for having me today. So last year, January, right before the pandemic, we had a workshop therapeutic antibodies, that large workshop, I presented antibody as a drug modality. So today I want to show you one example of how we generate antibodies for treatment of different diseases. Because we are still in the middle of the COVID-19 pandemic, so I want to show you one example that how our lab evolved in generating antibodies targeting COVID-19. So COVID-19 is a disease caused by the virus SARS-CoV-2. Most people actually got very mild clinical symptoms, but some people, particularly the elderly and people have comorbidities, have major severe symptoms, which also could lead to death. And as of today, globally, we have more than 100 million people infected, more than 2.6 million deaths. And in the U.S. alone, we have close to 30 million confirmed cases and over 500,000 deaths. So that's a pretty devastating disease. And the virus causing this SARS-CoV-2 is called SARS-CoV-2, which is a single-stranded RNA virus. In fact, actually, coronavirus is not new. So we have some circulating viruses in the human population, which causes very mild diseases, such as the human coronavirus, OC43 and Hong Kong 1. But in 2003, there was a major outbreak of SARS-CoV, and in 2012, there's a MERS-CoV pandemic. And of course, started in late 2019, we have the SARS-CoV-2, which is the COVID-19 pandemic. And so the virus has this spike protein that has a single-stranded RNA genome, and also have the envelope protein and the membrane protein, which is shown in blue. So how the virus works is the virus will attach to the host cell by engaging the host receptor ACE2. So once that happens, so the spike protein will mediate the viral fusion with the host membrane, that release the single-strand RNA genome inside the host, that hijacks the translational machinery of the host to replicate the viral genome. So in the process, actually, you need to have a couple of viral proteases need to cleave the transcribed messenger RNA into the different proteins. So therefore, we can generate protease inhibitors, small molecule inhibitors, as the potential therapies for blocking the viral infection. This is actually a very active area of drug discovery. And once the genome is during the transcription replication of the viral genome, so we can also design nucleoside analogs. For example, the drug remdesivir by Gilead is such a drug which can block viral genome replication. So the nucleoside analogs are another major drug candidates for blocking viral replication. So once the virus replicated with the code proteins are produced, then the new virus are generated that's secreted outside of the cells that can initiate new infection. So, another major drug discovery activity actually is using antibodies, monoclonal antibodies, that can block the viral engagement of the host receptor. So this is what I'm going to talk about today. So as of today, we already have two antibodies, one by Regeneron, one by Eli Lilly, approved for emergency use by the FDA. And there's another 5 antibodies in phase 3 clinical trials. Another thing that is, we already have drugs actually they made into these so-called cocktails. So they have 2 antibodies in a cocktail, which will provide a viral escape, which I will talk about this a bit later. In addition to these 7 late-stage phase 3 antibody drugs, we already have another 5 in phase 2 clinical trial, and 11 in phase 1, and hundreds in preclinical development. And the way we generate antibodies for COVID infections, there's multiple platforms. One is through the antibody phage library, which I'm going to talk about today, which is very quick. You can get antibodies in about 1-2 months, and can generate a very potent, fully humanized antibodies. And you do need a BL3 lab to do the screening, so that's convenient. And we can also use the humanized mice, which are producing fully human antibodies. An example of that is the Regeneron antibody. Another thing is that we can isolate antibodies from B cells infected with the virus that recovered from the infection. So these actually majority of the antibodies in clinical development generated such a, for example, the Regeneron antibody and the AstraZeneca as antibody as an example. Of course, we can also use the conventional method to immunize animals such as mouse, rabbit, even llama, which generate single domain antibodies. But this approach, animal immunization requires a humanization step, which takes about half a year. So given the urgent need of medicine for the COVID pandemic, this is a slower process. Example, this is antibody generated by at the University of Pittsburgh, which is also in clinical development. So very early, my lab, I think in February last year, as soon as we got the secrets of the virus from China, we generated the receptor binding domain of spike protein, which show you spike protein S1, S2 domains. You have a receptor binding domain in purple. So we produce this protein as efficacy fusion protein in 293 cells, show you here. And we also produce the original SARS-CoV RBD as a control. And as shown in this binding assay, with we can tell that the protein, recombinant protein produced in 293 cells, they can bind to the human SAE2 protein with high affinity, about 0.38 nanomolar. And in comparison, the original SARS-203 SARS virus, the RBD binding affinity to the SAE2 receptor is about 1 nanomolar, which is the current virus is about a 3-fold more potent in terms of binding to the receptor than the original SARS. This explains why the transmissibility of the current virus is so much more severe than the original SARS virus. Using the protein generated, I show you here, we paired our displayed human antibody libraries in our lab, and we recovered phage antibody clones that can bind to the SARS-CoV-2 receptor binding domain, in this case, we isolate about 149 positive phage. Over this 149 positive phage, we were able to get 35 unique antibody sequences, single chain FV fragments. From here, 35 we converted into full IgG one antibody 30. So from 149 to 35 to 30 antibodies. Using these 30 antibodies, we did use top panel binding to the IBD protein. You can tell that all 30 antibodies in red can bind to the IBD protein to a different degree. And of the 30 antibodies, only two, number 12 and number 20, can also bind to the original SARS-CoV IBD. So this is called cross-reactive antibodies. So, this is the IBD protein. We also tested the antibody binding to the host spike protein, which is a trimer actually in the viral surface. And the native prefusion spike protein trimer, you can tell also all 30 antibodies in red can bind to the spike protein to a different degree. And again, the two antibodies, number 12 and number 20, can cross-binding to the original 2 or 3 COVID virus. Then we did the neutralization assay. This is in collaboration with Pei Yu's lab at UTMB. So essentially, they engineered fully infectious SARS-CoV-2 virus with the reporter, amino-grade reporter. So if the virus, if the reporter virus can infect the cell, in this case, is a virus cell, is not human primate E6 cell, the cell turns gray. And if the antibody can neutralize the virus from infecting the host cell, you see the original color, no gray color. So show you here just one example. This is the mock infection, the control. And this is actually the control IgG, isotype control IgG. You say most of the cells get infected by the reporter virus. But one of the antibodies we generate, antibody number 6, can almost block the infection. So this is show you one example of this. So we tested all 30 antibodies, this neutralization in vitro cell-based neutralization assay. All 30 antibodies can neutralize to a different degree, but 11 of them give us 75% neutralization at antibody concentration of 10 microgram per ml. So that we focused on these 11 antibodies. So what I show you here is the top 5 antibodies. You can tell that the top 5 antibodies, they all can block the IBD binding to the human host receptor ACE2 protein in a dose-responsive manner. And here I show you the neutralization titer 50, UT50. You see all 5 antibodies show potent neutralization of the viral infection, the reported viral infection cell-based assay, which I showed you earlier. And actually these 5 all have UT50 below 1 microgram per ml. And the body affinity of these antibodies is a single-digit nanomolar range. And I also want to point to you that the most potent neutralizing antibody, number 6, is not necessarily the most high affinity body. Actually, this antibody has a 20 nanomolar, the lowest body affinity, but the highest neutralizing activity. That's why we need generate multiple antibody candidates to get the best antibody, which neutralization is the most important criteria for selecting potential drug candidate. So next, we investigated the binding residue of the antibody to the receptor binding protein. And in collaboration with the Integral Molecular, which is a company in Philadelphia, we prepared more than 400 mutant RBD proteins by replacing every amino acid residue in RBD receptor binding domain with alanine scanning. That these proteins expressed 293 cells as a membrane-bound protein. That we tested the binding of these antibodies to the different mutants. So if any residue with lost binding suggesting these residues are critical for the binding of the antibody. And by doing so, we mapped all these antibodies. I show you here two antibodies, antibody number 6 and antibody number 14. You can tell in gray, so the antibody number 6, this is the receptor binding domain of the virus protein. And we can tell that glycine 446 and K444, tyrosine 449, and T345. These are important residues for antibody 6 engagement. And antibody number 14, you have E484, F486, and this plus alanine 475 and phenylalanine 456. And you can tell these antibodies, they are not overlapping epitopes. They actually, two antibodies, they can bind to the same receptor protein at the same time. And look at another structure. So if you dock the ACE2, the host receptor protein, the RBD, viral RBD protein, you can tell that these antibody 446 and directly clash with the host receptor. And also in the same way, antibody F486 and E484 can also clash, directly contact with the ACE2 receptor protein. So this explains to you why these residues are critical for the neutralization activity of the antibodies. Of course, both antibodies can block the RBD protein binding to the host receptor ACE2 in a dose responsive manner. Another thing is that we know that if you treat the patient with a single antibody, the virus will generate escape mutants. So this is actually it is a concern. That's why I told you earlier that a company like Regeneron, Eli Lilly, AstraZeneca, they all produce the antibodies as a cocktail. So that's hope to provide viral escape. So in this study, so we did this. So we did a in vitro viral escape mutant assay. So we incubated the virus cell with antibodies, either two antibodies individually or in combination as a cocktail. And then we use 3-round selection. Each round we increase the antibody concentration, starting from 10 microgram per ml, and to the second round 20 microgram per ml, to the third round 200 microgram per ml. So you notice that after 3 rounds of screening, the cells infected with individual antibodies, treated with individual antibodies, generate escape mutants, but the cocktail did not. after sequencing the escape mutant, we show that antibody number 6 treated virus, they generate mutants mostly use a K444 residue. And antibody treated with number 14 generate mutants mostly 484 and 486. And let's go back here. You say 484 and 486 are the two antibody amino acid residues that directly contact the ACE2 receptor protein. And 444, which is the amino acid residue directly clash with the ACE2 protein for antibody number 6. So it's not a surprise that the escape mutants are mutated as those critical residues. As you can tell, with the mutation, the K444 mutated to R or arginine, so the antibody can no longer neutralize the escape mutant. But the antibody number 40 can still neutralize the mutant virus. Similarly, the mutant 484A generated by escaping antibody number 40 treatment can no longer neutralize by antibody 14, but antibody number 6 can neutralize. This actually explains why the cocktail antibodies the virus did not generate any escape mutants. So your panel say just explain to you that both antibody individually your cocktail can neutralize the wild type virus in a dose responsive manner, but the 444 mutation targeting antibody number 6 in red, then the antibody number 6 can no longer neutralize the 444 R or S mutant virus. Similarly, but the cocktail still can. And similarly, mutation in 484 and 486, which escaped antibody number 14 treatment, can no longer neutralize these mutants. Antibody number 6, you red can still. Cocktail, of course, can also. So this is a good example to show you that a cocktail you did good options for antibody treatment of COVID-19 infection to escape, to prevent neutralization and escape. And of course, we tested these antibodies, the efficacy in animal model. And in this vivo model, infection model, so we did is, so at hour 0, we charged the mice with the virus by nasal delivery. And in the prophylactic treatment model, so we added this sub-Q of the antibody 16 hours before the viral challenge, so this is a prophylactic model. Or in the treatment model, we added the antibody sub-Q 6 hours after the viral challenge. This is the treatment model. You can tell it is at 20 milligram per kilogram high dose level, the antibody number 6, actually number 6, and also antibody number 14. Number 6, actually, both the therapeutic and the prophylactic models, they can 100% prevent the viral infection, which is measured by a viral long viral load. And for at lower dose, 5 milligram per kilogram, the antibody number 6 can still neutralize the viral load by 4 log, and antibody number 14 by order of 3 logs. So the there's some issues related to antibody, IgG, VAR-based antibody therapies. So number one is actually the RV infusion of high-dose antibodies is costly and inconvenient in the clinic. And the second challenge that is the limited antiviral effect in the respiratory tract, because through RV infusion, only a small amount of the antibody actually can diffuse into the respiratory tract. That's where most of the infection takes place. And of course, IgG antibodies is a big issue with the viral escape generate resistance in the clinic. So for our program, instead of making another IgG VAR-based cocktail, we actually collaborated with a company in California called IgM Bioscience. We formulated the antibody actually as IgM. So these are 5 IgG molecules linked by the J chain. This is actually with high avidity. We formulated for nasal delivery antibody instead of RV infusion. So by doing so, the antibody we generated IgM40, which is drug candidate, can neutralize the US Washington strain. This is the wild type of virus. In red is the IgM. In black is the IgG version of the antibody. You can tell that the IgM version antibody in red is about 2 logs more potent, neutralizes the wild type US COVID virus. And more importantly, the IgM antibody is potently neutralizing the D7 spike mutation, which is the UK variant, which is actually became a dominant variant in UK and in Europe. Actually, the IgG antibody can also neutralize, but 2 logs less potent. More importantly, the IgM antibody is potently neutralizing the P1 variant, which is Brazil mutant, which is became a very dominant strain in South America, and also actually we also detected the mutant in the US. You say the IgG antibody can no longer potently neutralize the Brazil strain, but IgM can. Similarly, the B1351 spike mutant, which is the South Africa mutant, the IgM is about 3 logs more potent than the IgG version of the antibody. Just show you here. So by converting to IgM antibody, so these antibodies are 40-fold more potent than IgG in terms of the US strain, and 45-fold more potent than the UK variant, and 547-fold more potent than the Brazil variant, and 374-fold better than the IgG for the South Africa variant. So that we formulated antibody, IgM antibody as a nasal delivery, and you can tell that after 96 hours of a single nasal delivery, we see that most of the antibody still retains in the nasal cavity. Show you here. This is a live imaging. Show the labeled antibody still in nasal cavity. By its vivo imaging of different organs, we show that 24 hours, most antibodies either in nasal cavity, is lung, and 48 hours, same thing, is nasal cavity, lung. 96 hours, again, nasal cavity, lung. And 168 hours later, we still detect antibodies in nasal cavity and also in lung. You cannot see clearly, but you can see red there. And most importantly, this antibody works potently, can neutralize both prophylaxically and therapeutically in the animal model. Show you here that IgM antibody is a prophylactic model, which you give antibody 6 hours before the viral challenge as initial delivery. At 3.5 milligram per kilogram dosing, you almost say 100% block neutralization. At 1.2 milligram per kilogram, again, almost 100% efficacy. 0.4, again, 100% efficacy. These are 10 animals per group. And all the way to 0.044 mg per kg, a very low dose, we'll still see about a log of viral load reduction. This is the y-axis, you see the log viral load. So the control is about 5 milli viral particles per log. At the 0.044, we say about 400 solid. So that's about 1 log lower than the control group. Therapeutically, we still see efficacy at 0.4 milligram per kilogram, which is about 2 log below the control. And this is extremely potent efficacy by single dose initial delivery. And so just I don't have time to show you more this data. The paper the review for this program. I just want to the last slide to show you the summary of this IgM molecule. So the engineered IgM antibody, which has a high binding and neutralizing activity than the IgG1 molecule, which I showed you. And this the lead antibody IgM14 has 230-fold more potent neutralizing activity than the IgG14 for the wild type virus. The neutralizing activity of the engineered IgM antibody is determined by epitope of their parental IgG1 counterpart. Particularly, the epitope leads to the steric clash with ACE2. This is a magnetic explanation why the IgM antibody is so potent, which I did not have time to explain to you. So clinically, most important to say that IgM14, not the IgG counterpart, potently neutralizing all the major SARS-CoV-2 variant, including the UK strain, the South Africa, and the Brazil lineages. And in addition, the IgM14 antibody blocked SAE2 binding to a panel of 21 reported RBD mutant proteins, which I did not show you due to the limit of time. After initial delivery of the mice, IgM14 showed minimal effect dose is 0.044 milligram per kilogram for prophylactic treatment and 0.4 milligram per kilogram for therapeutic treatment. And IgM14 can be atomarized for international administration and exhibit desirable international pharmacokinetics and the safety profile in rodents. I did show you the data. And the clinical trial of this antibody starting next month. So this is truly a teamwork. So my lab is UTHealth, and Dr. Peyus's lab at UTMB, integral molecular, which did the epitope mapping of the antibodies, and IgM Bioscience, developing the antibody for clinical development. Shili Liu's lab at University Houston did the biodistribution in animal model, the study I showed you. And Susan Weiss' lab at UPenn did some of the earlier pseudoviral neutralization assay. And Jason McLean's lab at UTHealth did the structural work. With that, thank you for your attention. And let's try to answer some questions that we have.
Thank you, Zhiqiang. Great talk. So just open it up to the audience. Again, feel free to enter your question in the chat, or if you'd like, you're more than welcome to unmute yourself and ask your question directly. I'll start off with something that I was just curious about while you were talking, Zhiqiang. So with this IgM14, I think it's super impressive at how it seems to have activity against these mutant strains that have shown up. My question is, in the prophylactic studies, it was unclear to me how I saw 48 hours for the lung, how far those studies were carried out, and then thinking about what that marketing strategy would be. Would this be something that would be recommended to be taken by people along with the vaccine, or how would you envision that?
Yeah, so that's a very good question, Suzanne. So as you know, I didn't have time to show you the data. And the South Africa strain mutant actually already resisted to all the current FDA approved emergency use antibodies from Regeneron, from Lilly. So this is very concerning. And also show that the South Africa strain, also Brazil strains, showed less resistant to the vaccination. This is short data for the Moderna vaccine, also the AstraZeneca vaccine do not work as potently for those strains. And so with the pandemic going, so it takes about at least 1 to 2 years to immunize the whole world population. So it is conceivable that more mutants will be generated. So we need, in fact, already many companies like Pfizer, Moderna already making their next generation vaccine that covers some of these mutant strains. But unfortunately, the viral mutation is always a step ahead of the vaccine. So that's why we need have therapeutic options to control localized pandemic. And so that's in terms of the patient population for this antibody, we envision that they will be formulated as a nasal spray, like a nebulator, similar to like an asthma drug. And the IgM actually is about 1 year stable in room temperature. So anyone, so we use just a little bit of the antibody. You compare with IgG antibody, we need about 8 grams infusion. For nasal spray, we need milligrams of the antibody per spray, which last in mouse studies we already tested in about a week. So so what you can do is you can anyone just carry such a nebulator in your pocket, and every 1 to 2 weeks you can spray once as a chemical mask. So nice about this antibody so far is resistant to all the mutants we tested, and so for first time we may have a drug that can use prophylactically, because for the IgG antibody it's difficult actually to give it prophylactically, because it's in the hospital, the infusion is too expensive, too inconvenient to do.
Thank you. So we have a question from Stan Wadowich, who compliments you on both of you on your presentations. Looks like we've got a few coming in. Can you both comment on the use of other types of antibodies like single chain, other species? Can you comment on the use of ADC to treat other diseases? Might this be effective against autoimmune diseases? So I guess we could start with Kyoji first on this one.
Yes, technically speaking, it's pretty reasonable to use other delivery vehicles other than mAbs as well. The major reason why we focus on mAb-based delivery is its really long circulation life in the body. Other proteins such as scFv, single chain antibodies, and smaller fragments show really short circulation life, and it's pretty easily, quickly clear. So it's sometimes advantageous for short-term applications such as diagnosis or imaging, but for therapeutic applications, based on our experience, the long circulation life positively affects the overall therapeutic efficacy. So that's the primary reason why we prefer to use mAb to validate our linker technologies, or even Dr. An's therapeutic molecules. But again, yeah, technically speaking, it's not difficult at all to transfer the technologies we have developed to the other fields using other protein vehicles.
Yeah, I just want to add for other diseases, so there's a drug modality called AAC, the antibiotic conjugate. So the one example is Genentech has a drug candidate in clinical studies for treatment of drug-resistant Staph aureus infection, and that is you conjugate antibiotic that's very potent against the Staph aureus, and using antibody can recognize Staph aureus receptor, and that delivers antibody through internalization. Once you inside the bacteria, then proteolytically the antibiotic is released. It's very similar to killing a cancer cell. So that's one example. I really like that strategy because so that's one example. In terms of autoimmune diseases, I cannot think of one. Kyoji maybe also say Jing Wang from Baylor is Erodius. He's also expert in ADC. Maybe he can also comment. Are there any examples of ADC for autoimmune diseases? But I can think of for infectious diseases, and of course mostly for
Pfizer is actually trying to develop antibody drug conjugates for autoimmune diseases, though it's still in its infancy. Some papers have been published in bioconjugate chemistry journal. They attached a steroid type payload that has very high, unusually high potency, and that's why they couldn't use it as a single agent. But after attaching that to an antibody, it could show target-specific, reduction of the signals associated with autoimmune diseases. Though it's just a model validation, so it's still on its way for future development. But yes, some researchers have tried that kind of transition from oncology to non-oncology application.
Yeah. So that's correct. So I was thinking about are there any drugs in clinical development targeting autoimmune disease? I don't think so yet. But yes, in research, in academia, ADC is used in all kinds of applications.
And just to follow up on your comment, thank you both. Just if I follow up on your comment with the bacterial or antibiotic conjugated monoclonals, the same strategy obviously could be used for targeting COVID infected cells, right? Where so the approach you presented was looking at neutralizing antibodies, but obviously if one was targeting just the spike protein on an infected cell and delivering a cytotoxic payload, the idea there would be to try to kill any infected cell, because obviously once the virus is in the cell, a lot of the spread is from cell to cell contact as opposed to the virus budding out and being in the circulating system. So is there work being done to essentially to deliver a cytotoxic payload to infected cells?
Yeah. So that's a good question. So the idea is, I just want to elaborate what you said is that you can. So once the host cell is infected by the virus, this host cells are decorated by viral proteins, such as like a spike protein or some other proteins, a membrane bar protein, but just by randomly. So these cells actually are, so to speak, marked by the viral protein. You can imagine that you can get the antibody that recognizes the viral protein, and then you have a drug conjugate to that antibody can deliver to kill the infected cells. And so I do think people are developing such a drug for antivirals. The fact that really is because there's other antibody options, better options, that is through bispecific. So what people do is actually in my lab we do a lot of this developing bispecific antibodies targeting viral infected cells. The idea is that is you have a one antibody targeting the virus that's a viral protein that's decorated the infected cell. And another antibody actually recognizes the T cell, let's say CD3 T cell. So the bispecific antibody will bring the T cell in proximity to the infected cell. It's a much more effective way to kill the infected cells. And in fact, this is actually a therapy. This actually we used very widely in cancer treatment called BITE. Your comment about the ADC to kill infected cells are valid. So I myself have not seen any publication yet, but clearly that's possible.
Okay, thanks. We've got a question here from Ritu Gupta, who also thanks you and asks, can the IgM antibodies provide long-term or short-term protection?
Yeah, the actually based on the animal models, so we, like Suzanne, you said, is 48 hours. We did the long viral load, so that's the time point we can say. And the really is after 40 hours, the control group will be loaded with the virus, so there's no so based on the biodistribution, we can say that the single dose delivery, initial delivery of the IgM antibody can stay as long as the nasal cavity for up to 168 hours. So that's close to a week. And based on the high potency of the antibody, because IgM actually they are secreted in the nasal cavity as our natural mucosal immunity. So they are naturally very sticky in the nasal cavity and in the lung. And so we have to do the clinical trials. And our hope is that the IgM antibody will provide the long-term protection up to say 2 weeks, based on the half-life of the antibody in the nasal cavity. Clearly, this needs to be tested in the clinic, and that's our hope that will provide. In terms of, if you talk about long-term protection, in terms of like vaccine protection, no, the IgM is still a therapeutic. It's only effective when you have enough antibody in the respiratory tract. So I would say up to 2 weeks. That's our hope.
Okay, thanks. So here's one for Kyoji. What accounts for the 14-day plasma stability of circulating ADC? I would have thought that the ADC would eventually enter cells and be degraded, also from Stan Wadowich.
Okay, yeah, the data I showed is the circulation life of ADC injected into healthy mice, not in the tumor-bearing mice, and the amount of ADC in the blood sample was determined based on the conjugated payload rather than the mAbs structure. So using sandwich ELISA method, we quantified how many the conjugated payload molecules were still attached to the antibody. So this way you can quantify the loss or the retention of the conjugated payload rather than just the clearance of the entire antibody drug conjugate structure. So regardless of the type of linker, we can see standard 2 to 3 week half-life of half-lives in all ADC groups. But if you quantify the conjugated, intact, non-cleaved, ADC payload, you can see that kind of clear difference between labile linker-based system and our stable linker alternative. Did I answer this question properly?
Yes, thank you.
So following up on that, are there linkers that prevent warhead efflux from the cell?
So that from the chat.
Yeah, are there linkers that prevent efflux of the warheads from the cell?
Oh yeah, that's really a great question. Actually, modifying the payload part is one of pretty exciting topics because most payloads, ADC payloads, are more or less hydrophobic, and they tend to be cleared or pumped out from the cell. Though some payloads are very hydrophilic, and it's not a problem. But once you try to modify the polarity of each payload molecule, that sometimes nullifies its cytotoxic activity because of the loss of proper interactions with its target molecules. So fine-tuning is really important and key to get still, potent and more preferable new payload classes. It's a very challenging topic. Many medicinal chemists are working really hard to fine-tune the molecular properties, so they don't compromise its original site killing mechanisms and interactions with its target, but increasing the polarity to prevent that efflux. Our lab is not working on that part, but yes, that's another thing. They'll come up from some of the labs in the near future as a toward more promising payloads.
Thank you. So we've got a couple more here. I know we're running out of time, but Naveed Sabani compliments you both and asks if you can comment on peptide conjugated to drugs.
So this means instead of mAb, what about drugs conjugated to peptides? Yeah, that's also another hot topic, that have been pursued by many other labs. But again, as I explained to the first question, the biggest concern is the circulation life. So most peptides are cleared within 2 to 3 hours from the body. So that means very frequent administration will be required to implement effective treatment. So it depends on what you want to do with whatever agents you want to use or in your hand.
Okay, and then our last question in the chat is from Jin Wang. Great talk, Zhiqiang and Kyoji. Kyoji, the EVC linker, does the acid residue slow down the linker cleavage by cathepsins compared with VC?
Yes, thank you for that very detailed technical questions. Actually, it was our surprise. Addition of E rather promoted the degradation or cleavage by cathepsins compared to conventional valence citrulline. So E not only just prevent the premature degradation in mouse and human plasma, but also promotes cathepsin-mediated cleavage. We haven't got any data supporting that increased cleavage rate improved the overall efficacy in vivo. We are still trying to figure out if that is actually critical or not. But this finding may be important in for other applications as well, such as probe design to quantify the cathepsin activity in cells or in some of the models. So I believe this finding is also really interesting for such kind of other applications as well. Okay, well, we are just past 5 o'clock. I want to thank you both again for fantastic talks. Lots of really exciting data. I'm looking forward to hearing about some of these clinical studies that are getting ready to start up. Yep.
Yes,
and yeah, it should be exciting. So thank you both, and I want to thank everyone in the audience for attending.
Yeah.
Thank you, everyone.
Thank you, Suzanne, for organizing.
Bye-bye.
Bye-bye.